We are Gonzo35, and today we will be providing you with an informational video regarding translation. Overall, translation occurs in the ribosome, a large multi-protein complex that interacts with mRNA and then assembles a polypeptide chain. The small subunit binds to mRNA and recruits the large subunit at the first 5' AUG or start codon with its associated tRNA.
Translation starts with mature mRNA. It is recognized as mature mRNA in eukaryotes as having a 5' cap and a 3' poly A tail, which help protect hydrolysis by enzymes of the two ends of mRNA.
The 5' cap also helps ribosomes recognize the 5' end, which is needed for the ribosome to be translated correctly from the 5' to the 3' end. It also aids in the export of the mRNA from the nucleus. The 3' poly A tail contains 50 to 250 adenine nucleotides. It also aids in nuclear export, and it stabilizes the mRNA on the ribosome for translation.
DNA encodes for protein sequence information using 3-letter codons. This is known as the degenerate code, meaning that there is more than one codon per amino acid. There are 3 stop codons, which do not have amino acids associated with them. The ribosome can start translating the mRNA once it recognizes the correct reading frame. Establishing this correctly will ensure the correct polypeptide. In prokaryotes, the Shine-Dalgarno sequence recruits ribosomes to the start codon, while the eukaryotes have a ribosome use the first 5' AUG codon as the start codon. This whole process always uses codons on the mRNA, going from 5' to 3' to make the protein.
The very first process of translation is initiation. In this process, it occurs at the 5' AUG or start codon. A small subunit binds to mRNA near the 5' end and basically scans for the first AUG, meaning start. The large subunit can only assemble on the small subunit, or the mRNA around a met tRNA. This initiation creates the initiation complex, but it also needs the small subunit with met lined up first. tRNA does the job of translation within the ribosome, and it looks somewhat like a clover leaf. In the ribosome, we have 3 sites: the E site, the P site, and the A site. The E site is where the polypeptide chain exits the ribosome. The P site is where the new polypeptide chain actually forms, and the A site is where the new amino acid comes in.
The process of elongation begins with the ribosome reading the mRNA's message beginning with the start codon AUG. The ribosome brings in an amino acid tRNA into the P site with a complementary anticodon to the start codon. Following this, another tRNA is brought into the A site with a complementary anticodon to the following codon on the mRNA. The anticodon goes from 3' to 5' in an anti-parallel manner to the mRNA, which goes from 5' to 3'. The tRNA is fixed in place through the hydrolysis of GTP. Next, the bond between the amino acid and the amino acid tRNA is broken into the P site, and the amino acid forms a bond with the amino acid of the tRNA in the A site simultaneously. The ribosome moves 3 bases down the mRNA, and another tRNA is added. This process continues until a stop codon is reached.
In the process of translation occurring right after elongation, there is a process called termination. This is where a stop codon is hit, and a small protein called a release factor interacts with RNA at the stop codon. It jams itself into the A site, and the polypeptide chain is cut free. Stop codons include UAG, UAA, and UGA. The role of a release factor is to stop the codon in the A site. If we weren't able to have a release factor, the codon would continue on and we wouldn't be able to stop the process of translation.